I'm is this working? Yeah. Okay. All right. So, hey everyone. So we'll be discussing today protein structure prediction, like from classical methods to AlphaFold 2. So we'll talk more about to what's AlphaFold 2, and I totally love this topic actually. Like while reading through the some of the papers, watching lectures, and why this is important. So we have been reading more about the proteins, uh, but not not talking about more about structures. The structure looks like it's the most important part in the proteins. Alpha fold. Oh yeah, yeah. Actually, when I tried like the Google Slides grammar checker said it's two words. So
Yeah, it's a suggestion
yeah,
from them, right?
yeah,
You
yeah,
don't
yeah.
have to listen to them.
Yeah, yeah, yeah. Um, all right. So why are we talking about this? So the 2024 the Nobel Prize for Chemistry was awarded to these three people. So the first is David Becker, like who is a very famous uh, bioinformatics scientist who has been working for decades now on this, and who has designed the Rosetta software, which we'll discuss in very brief. And the person with glasses is Damis Hassabis, and he is the lead of Google DeepMind. So he was the architect of basically like how. So he was basically a gamer, and so when David, so basically, it was very difficult to find these structures from the protein. So the David Baker basically designed a game that how could people come and play with and find the most optimum structure to beat the gas leaderboard, and that fascinated Damus Hassabis, and Damus Hassabis, inspired from that, uh, took a team in Google and led the revolution of AlphaFold and AlphaFold 2. So we'll read more about that, and so we'll look into more details. So what made it different from the classical methods and all of this. Right. So so these are the contents, like what are protein and amino acids. So why predicting protein structure is so hard. So what are the pre-deep learning approaches, and what is AlphaFold? AlphaFold 2. What does it do? How does it work? And uh, and I tried some of the simulations from AlphaFold to a labs notebook. So we'll also try to see. And but when so it's very fascinating. But what uh, what are its drawbacks and when it does not work? Okay. So why are proteins important to us is basically so ligaments and arteries. So they are made up of proteins, and basically they help us understand like what drugs can help us basically treat them and all of these. Um, this, and So coming to amino acids and protein, so so protein is basically a combination of a sequence of amino acids, right? And but so in the sequence, so there are 20 types of amino acids, and the molecules between these different amino acids interact with them to form structures. So it looks like, okay, so there is a sequence of amino acids, but in nature, the molecules interact among each other to form peptide bonds, and that comes up as a structure, right? Um, so, so when we talk about structure, so there are four levels of structure. So one is the very simple, like the primary protein structure is the sequence. The secondary protein structures are like the alpha helix, and so these are the primary structures, and there are more complex structures, tertiary structures, and the quaternary protein structures. So these are the four levels of protein structures. Uh, yeah, and so what is the problem definition here? So the problem definition is like given an amino acid. So how do we come up with the 3D structure of the protein, right? And so uh, so when we have a sequence, so it can take many structures, but we are looking for the most stable 3D structures, right? Okay. Uh, so by the energy of the molecule is at the lowest. Uh, right. Yeah. Uh, so so actually, I'm not a a bio guy, so I'm from. So it's very difficult to understand this bio concepts and trying to relate all of these. Uh, so how are these predictive structures used? So it's basically it can be used in drug development and predicting the function of a new protein. So most of the people are allergic to some kind of drugs. So we can basically synthesize those drugs based on this information, and basically we can explain how protein functions and how one might uh, alter with its function. And yeah, okay. So so there were so why not just solve structures experimentally, right? So what this means is basically that so some of the scientists, what they try to do is like they try to take a mass of protein, or like which let's say my this muscle mass has some of the proteins. So they try to put that in a X-ray crystallography, and they try to then model that okay, so these are the chemicals, and these are the chemical bonds. And but the problem with that is like it's very expensive. It costs $1,000. And uh, and the total number of and there is a very huge gap between the total number of sequences and the total number of structures, and this gap is widening because we are getting more number of protein sequences, and the number of protein structures we know are very limited, right? So yeah. So the total number of structures are basically like some of the structures we know, right? Like the primary structures, but these like the templates that we will see. So uh, yeah. So basically there are four levels of protein structure. So that we discuss is like uh, that a protein is basically a combination of amino acids, and they interact to form these structures. And we are trying to find the most stable structure. And yeah, the first is the primary, like which is a sequence, and second is the like the most basic level, the alpha helix and all of these. So these are the protein structures, but not necessarily that the protein will have uh, uh, these exact copies of this. So it can have combinations and various.
Yeah, but DNA, RNA, then you have a protein. So DNA is the code, the recipe to make the food. Protein is the food itself. Yeah. So if you change something in the genetic code, the protein will be might be different. Oh, yeah. That's the thing we see as phenotype. For example, a disease. So the disease, what you see, what you observe, is the protein. So protein mixed. Hair color is different, color and so on. It might be related to genetic variation, it might be related to something how these proteins bind to each other. So based on the structure you can see from here, you can tell what's happening. The method or mutations and everything changes into the name. Will be translated into protein. Oh yeah. But not necessarily. There are non-coding things will not be translated. So even if you change this region, you won't get any change in the protein. There are coding regions. Something the protein will be. Oh yeah.
Right. So thank you for the explanation. So why not just simulate the folding process? So we know the sequence of molecule. So we can basically simulate. Okay, so this molecule will react this way, and apply the laws of physics. But looks like so it works for very simple proteins. So here you can see that uh, the blue is got the blue structure is got by the simulation, and the red structure is got like from the experiment. And you can see that they are similar, so they are not very distant. But uh, but these proteins are very simple, and are not uh, uh, like very difficult to modulate. And also these experiments take a lot of time, so that's why we need better learning algorithms that can basically learn this 3D structure. So yeah, so for most proteins this doesn't work because folding time scales are usually much longer. And I saw a video which said that so even if we have uh, very powerful supercomputers, it would take 200 astronomical years to predict some of the proteins. So and also the molecular mechanics, the physics that we know between some of the molecules is not very accurate, and and some of them are very hard to mimic the simulation. So so can we find simpler physics-based rules that predict protein structure? So for example, can we look at patterns of hydrophobic, hydrophilic, or charged amino acids? And people have tried all of these, but there is no success. So can we uh, try some knowledge-based methods? Some knowledge-based methods like, okay, so we have a protein data bank, so which has some proteins and some of the structures. So can we try some learning-based algorithms, some data-driven um, um, algorithms on these proteins to basically learn the uh, structure of these information? So yeah, so 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 let's see at some of the algorithms. So but so before applying some learning algorithms, so we have to know that uh, so proteins with similar sequences can have similar structures. So then only we can have a learning algorithm that can learn from this hypothesis and uh, try to predict the structures of some of the new proteins. So proteins with similar sequences tend to be homologs, meaning that they evolved from a common ancestor, and the fold of the protein tends to be conserved during the evaluation. And even proteins with 15% sequence identity usually have similar structures. And yeah.
Previous one.
Oh, yeah. Actually, I try to change the order later, but uh, yeah. Uh, so for most human protein sequences, we can find a homolog with no structure. So so you can see here that the plot shows uh, the the fraction of amino acids in human proteins that can be mapped to similar sequences in the protein data bank. And so you can see that so the uh, color of the shade of the blue basically shows that how many percent of the proteins that we know exist in the protein data bank. And so what is template based structure prediction is basically uh, so we have a query sequence which structure is unknown, and we search the protein data bank with a similar sequence and known structure and pick the best match. So that's a template. And so once I get the template, I start building upon it to come closer uh, to my query protein. But what does it mean for two sequences to be similar? So do I count only the number of amino acids that are similar? So yes. So I can see the sequence identity, the amino acids that match exactly between the two sequences. So then I can have a template from the protein data bank and I can develop on that. But can I have better similarity matrices metrics? So yeah, so I can have one more matrix. So these two are glutamic acid and aspartic acid. So they have similar behavior. So I can also have that as a similarity metric measure. So yeah, so this is because
What
like
do you mean by behavior?
so by behavior uh, so by behavior is like they have similar answer to some of the uh, chemicals and all of these. Uh, so like so instead of just comparing amino acids, so can I take some cues from the experiments or like how the reactions to other molecules and take that as a cue to do the similarity search. So that's the thing for the template. So if so earlier I used to do just amino acid search, like okay these amino acids, then I can take this template. Yeah, but can I have more like
How do you react?
how to react with it? So can I take that information and also come up with a better template? So, but so can we do even better? So once we identify some homologs to a query system, so we can basically uh, uh, so we can basically use this homologs to basically design the protein structures, and then basically we can use this hidden Markov models to basically learn the protein structure. Uh, so this is also kind of template-based modeling ab initio structure prediction, also known as de novo structure prediction. So these were kind of like template-based modeling. Okay, so come up with a template from the search database and then develop on it. So this was the pre deep learning era, like so where there was no deep learning algorithms used. So most of the models, so one of the models that was used was HMM, hidden Markov model, and now so this was also the Rosetta software, which was basically designed by um uh, David Becker. So basically, uh, so they basically had a physics based simulator, which which they call the knowledge based energy function. So basically, they fit in some rules like. Uh, and they call it the Rosetta energy function. So Rosetta energy function is basically they try to model the physics by knowing some of the rules. But when in deep learning, what has happened is like they gave the molecules and they gave the structures, and the model had to learn the physics by itself. So, so yeah, so these were the key ideas, and now let's see the AlphaFold. Uh, so what changed in AlphaFold is basically uh, so so when Google took over this project, so they had a lot of GPUs, and they wanted to use some deep learning algorithms for predicting these 3D structures, and so this is basically a transformer based. So this was the first project like where basically they wanted to use transformers. And so in template based modeling, what we try to do was like we try to search for one template that was the closest and develop on that. But in transformer in Evo, so basically what they called was Evo format. So there was multiple sequence alignment. So there are so these type of architectures are called two tower. So two tower means both towers process different types of context. So we see here that so one is the genetic database and another is the structure database. So one tower processes and tries to find out relationship between different genetic sequences, and the other tries to find out the relation the context between the various structures. And uh, and these two structures, so through the evo former also interact with them, they exchange information. Okay, so this structure is not so the geometry structure says that okay, so this structure is not does not obey the rules of physics, and so these context helps the sequence alignment find better sequence and so so yeah, so we have the data now and we have a new architecture which is transformer based. And uh, and the major technique that was that helped
First, the input sequence
protein, yeah, that's a protein
protein
sequence.
sequence.
Yeah.
Then you align it to it.
uh, uh, so we align it with the final 3D structure.
No, first the MSA. What is MSA?
Yeah. So MSA is multiple sequence alignment. So what we try to do is like so there are two search. So one is structure database search and the genetic database search. So in genetic database search, what I'll have is like I'll search the top the protein sequences that are similar. And in geometry based search, like what I'll try to do is like I'll take the top k geometries that I search, and then I will try to feed into the model. And finally,
you align sequence or the sequences that they try to get
yeah,
the one with the highest similarity
yeah,
with the inputs.
yeah,
Okay, and you got the group of those similar,
yeah.
and then what you do?
So then I try to see that, and this is my output series, right? So what the model tries to do is it tries to learn that how I can come up with a new structure that uh, uh, that is originating from that protein sequence, and that is also grounded with the genetic sequences and the geometry structures. Yeah. So in earlier works, we had two things. So one was the physics solver and one was the template modeling. And so we choose a template and we give to the physics solver, and the physics solver tries to simulate it. But here, there is no physics solver. So the assumption is that the model is trying to learn the physics of the molecules in the data-driven way. So that's also one of the disadvantages, that we don't know how this model works. So it's not interpretable. We throw some data and it gives some output. So one of the disadvantages also is like does it memorize? Okay, so we have given some data, and does it memorize? So I also have that graph. Uh, yeah, so Evo former. So basically, so Evo is basically coming from the evolution, and former is coming from the transformer. And so why evolution is basically, since they give a genetic database search, the model is trying to understand that how the protein sequence or the structure has evolved from some similar sequences using transformer. Yeah. And so there are some concepts like high confidence and low confidence, which I'll try to explain in the slides next. So the key ingredient is multiple sequence alignment, right? So so evolution is basically like how we can use the existing genetic sequences to predict the uh, from the database to predict the new sequences. And so this is the multiple sequence alignment, that was the novel part. And so this is the basically a guided search in a good energy function. So what basically says is that so this is uh, so this is basically so this is a picture from their paper, and what they basically say is that uh, uh, MSA helps to learn a good initial guess. So MSA helps to learn a structure which has a lowest point in energy. So blue to red is basically, it shows the amount of energy uh, that so if the structure is unstable, so it's likely to have higher energy, and if a structure is stable, it's likely to have a lower energy. So it shows that how the multiple sequence alignment provides a good initial guess, like a good template on which the model can develop. And uh, yeah, so it basically learns the uh, its objective is to predict a good structure for that protein.
Does it do anything? Is it
Yeah, it's a deep learning technique. Yeah, it's a transformer based thing.
also the MSA is
So MSA is so since we feed multiple sequences, and the transformer layers try to process the multiple sequences through their attention based mechanism.
oh was it the transformer or
Yeah,
was
yeah, it was
like
yeah.
faster?
Uh, so I was kind of confused, like which part they called MSA. So I thought, like, since the transformer learns, the complete mechanism is called.
MSA is similar,
Yeah,
but some of it doing deep learning.
yeah.
Some of it, especially hidden mark model and stuff. But the evoformer is not part of the MSA.
Yeah, I think because it learns the, so I think MSA is
Learn structure from sequence. Doesn't do any other.
right.
Provide bunch of sequences, give you the projected structure.
So so doesn't so alignment is only between the sequences, but there is also structure, right? So there are two towers, like the structure and the.
I'm not aware of aligning structure. Maybe use MSA for aligning the structure.
So MSA is used for so there are two so MSA is used to get the top k sequences, and the deep learning algorithm is used to.
Learn the structure for DC.
Yes,
Then they compare those structures
yes,
together and
yes,
they try to predict what is common.
yes, yes, yeah. So can we say that deep learning is part of aligning structures but not the sequence, right?
Predicting structure.
Yeah, okay, yeah.
But you could still use deep learning for alignment, deep learning for comparing the structure. That's part of the algorithm. But for learning part, not sure. If next time you provide a sequence, it tells you that this is similar to this and this without doing the actual one.
Yeah, it can't do this. So this is also a disadvantage. Like, so we cannot sample from the model that, okay, so these are the top k similar. So that was a disadvantage that was mentioned in one of the review papers.
Okay, so they are doing real MSA or they are learning. Who's aligned?
They are not learning.
Okay.
Yeah, because we can't. So let's say the model is trained, so I can't use that to uh, do sequence alignment. So I can only use the model to predict structures.
Like that they are similar versatile, they are similar and versatile, but it
Some of them they use.
uses some guesses. And okay, you like like represent those like using these some traditional approach of dynamic programming or something else is
So those dynamic programming would fail because it works on pairs.
It's not my
Here, MSA is 10 sequences. You want to compare them to a single sequence. That's why it's called multiple flat. So in the very old days, they were using dynamic programming. Very expensive. For every pair, so you take one and one, compare them, and then you take the results, you compare to another, you keep doing in pairs. That's very expensive output. So recently, they start doing like hidden mapping. They build a graph, and based on the um, character in each column, they try to align them. So they shift the node based on whether all the character in one column are similar or not. And they keep building that graph based on all sequences, not pairs. This is how they do it, sequence alignment for all of them in the same thing. You can think about it as a graph WFA, for example. Yeah, you can use it. But some of them still using traditional method, not related to machine learning. That's why I was asking him whether they were real learning or was really like actual comparison between the sequences.
got it. So I think the transformer learns the MSA, some part of MSA, not exactly,
Okay.
because so there is a K cross K matrix which learns the similarity between the K sequences, and there is a K cross K similarity between K structures. So I think those help it learn. But since the objective is to predict structure and not sequence alignment, it learns some part of sequence alignment, I think. So I will have,
Maybe, I'm not sure.
yeah, I'll have to read
You read the paper, right?
yeah, in this paper they don't talk about this. So
No, but there is a lot of
yeah,
nothing.
yeah. So all right. So how well does it really do? So there are a lot of work that say that the AlphaFold 2 is really good, and after this, so we have around 200 million protein sequences, but we don't really know yet uh, because the accuracy will depends on the quality and depth of multiple sequence alignment, and and also large machine learning models might memorize some of the training data. But it looks like AF2 was not overtrained. But yeah, I think we need to keep an eye on this. What by overtrain I mean memorize. So I have some protein database and some structures. So if I overtrain on those sequences, so can it still perform better on test? So I don't want the model to memorize. I just want the model to find uh, similarity and learn the structure so that I can use it. I can use those learned physics concepts in predicting new structures of the proteins. So yeah, so how well so AlphaFold 2 was the turning point in predicting structures because so there was a leaderboard CAS 14 and AlphaFold 2 was the first model which basically got 90% of the sequences right, and you can see also here the distance between the learned structure and the original structure. So how there was a drastic gap between um, the uh, the learned model's distance and the previous model distance. So
Wait a minute. Can you go back? What is the x-axis?
yeah, so the x-axis is basically like the various models that earlier existed, and
Year or
so it's actually basically the model.
model name?
Yeah, and the y-axis is the z score between the of the CAS 14 sets basically, and they show that how AlphaFold 2 was the winner.
I see.
And these two are basically the So these are CAS 14 scores, and these are the distance in Armstrong of the. So this is basically of the carbon molecules, and these are of the all the molecules. So the lower the better, and this one the higher the better. And so the same authors also did an analysis like, is it just memorizing the PDB or doing the homology modeling? So from this graph, so we can say that uh, that uh, the performance of novel structures uh, suggests that the model is not memorizing the data. That the model is not memorizing these structures, but instead it tries to predict the structures. So these are on the novel protein sequences on which the model was not trained. So yeah, so this was an important part. So when can you trust the prediction? So this is kind of a reliability diagram. So in a machine learning model, also like to know that how well it is, how good a machine learning model, the accuracy of the model should be equal to the should be linear to the confidence of the model. So, uh, so, so if the PLDT is so this is basically the distance metric. If the distance between a distance metric is between 0 to 50, the model is very unsure, and if the model is very blue, then it's very sure. So, so we can see that how so when to trust those predictions. So let's see this slide. So this is the structure of a protein where this model is trying to predict. The red regions are basically where the model is unsure, and the blue regions are where the model is highly confident about the structure. Uh, so there is also another interesting study that this, since we now know that AlphaFold 2 is good in predicting the uh, uncertainty of a structure, so we can also use that to know whether whether an existing predictive structure in the database is disordered or correct. So this was the plot. So you can see that how between PDB unresolved and human, so there is a similarity. So most of the structures which the AlphaFold 2 was not able to predict. So it shows that the humans were also aligned with the AlphaFold 2, that okay, these structures are not stable, or like we don't know their structures yet. Uh, all right. So there is a collab notebook, collab fold, like which I was able to run. And it's pretty easy, like bug free, and I was able to learn. Oh, uh, so actually, have some 3D structures with like which I couldn't put in the slide, but I was able to run very quickly on collab. Actually, I have the link here. Uh, okay, so I think this will try to get a new session and new collab session, and I can't show, but I can show in laptop. I think they are still there. Oh, right. Yeah. Uh, okay. All right. So I'm pretty much over the time. Yeah, I think yeah I've put here. So these are the structures that I try to predict, and I like you can also get these structures in the collab. It's free for running, and it's very quick. Uh, yeah, yeah. So basically, N to C, the colored by N to C. So N is basically the start.
It was lucky to run it online. Then
Yeah,
it's installed.
yeah. So yeah, and all right. So so what we don't have yet is like it's very expensive to train AlphaFold 2, and so we cannot use as a sampling engine or energy function. Uh, like we can only use it for generating structures and not know that how a good structure is. And yeah, so we can't basically change the protein structure after it's created. And so those things are, I think, currently people are working on, and we don't have any insights into protein folding. So why does protein fold this way and all? So the model is really good, but it can't explain. And yeah, thanks for everyone. No. So I have only tried AlphaFold 2, and so there were many variations for AlphaFold 2 also, like multiple sequence alignment techniques. And so yeah, I was only able to try those. Other tools, as in? No, like AlphaFold 2, yeah, AlphaFold and AlphaFold 2. Yeah, and so I was also looking into the RF diffusion, like which was made by David Baker. So I was also trying to run that, but um, I'll try to just maybe.
So in the collab, what do you provide the sequence?
Yeah, we provide the sequence, and it searches over the using the multiple sequence alignment algorithm. It basically gets the top key sequence, get the top key structures, and it tries to uh, uh, get the structure through the 5 iterations.
So the final output,
Yep.
does it give you any indication?
No.
No other information.
So there is another information, like how many sequences it uses. Like so for the one that I uses, it uses 8,000 sequences at once.
Build the structure.
Yeah,
Or to protect.
to predict the structure. And yeah, it also says how confident it is on that structure. And
Does it tell you any?
no.
And from the color scale, you can know what part is
Yeah.
for sure.
Yeah.
What part is very.
Yes. Yes.
Right. Any question?
I have a lot of questions too, but actually this is really interesting, like how AlphaFold 2 changed a lot of things, and how and it was also fascinating to see that how they made everything collab. You can also try for new structures that you can get more deep insight into what's not working and what's working. And yeah, and
I remember like 5 years ago, it was a pain. There was an initiative that download the software in your machine. Your machine will be part of the sharing ability like that. Anyone would like to collect protein, you have like 5,000 computer connected to the network, and we use some resource out of all these machines. You can be part of that network. This is how it was in the old days. It was really requiring a lot of resources, a lot of GPUs, a lot of hardware. Once this model out there, out of Google, Microsoft tried to do something. Then no longer you need to sacrifice your machine. They have a lot of, but it's not only about the hardware. Algorithm, as you can see, is really part of this thing. Make very smart algorithm that does not require that much of resources, plus the accuracy that you mentioned. So all of the existing model were less than 90%. That was the only thing jumping that ratio up. But every once in a while, like every 6 months or 1 year, they issue totally model. So it's going really very fast. Once one of those companies being responsible for development and so on, better than the open source or the community effort, much faster. Because of course, given the scale of the server they have, ability to search against It is. Some of the companies try to compete either by accelerating it, making just faster, or compete in terms of accuracy. But still, you cannot. It's really hard to compete with companies. You can improve a little bit, but by the time you improve something, they will be releasing a new model. It's you will lose the race.